Welcome, Chem 114 students. Carbohydrates, sugars, things that we take in our diet that help provide energy. And we'll learn, although we're in the second part of the organic chemistry course, we also will go into the third part with biological chemistry because carbohydrates are very important to our body system and the energy that we need to extract from foods. We can store energy as well in what they call glycogen, and we can also use energy sources such as direct sugars to get immediate energy. And there's a connection between sugars and carbohydrates and the TCA cycle and the citric acid cycle, glycolysis, and then of course comes the TCA cycle. Glycolysis is going on in all our cells almost all the time, and we then have the TCA cycle that goes on in our mitochondria, which is a very compartmentalized organelle. So let's begin. Um, Glucose blood levels, very important. People have to have sometimes deregulated blood glucose levels that can be very challenging, either too high or too low, can be devastating. So we have people who are experts in that field. You, as a student, may eventually become an expert in managing diabetes and, of course, blood glucose level. And we'll look at the glucose molecule, which is an aldehyde, and we'll learn that glucose is an aldehyde and it's an aldose. And we've talked about aldehydes because that's really the primer to getting into sugars, along with ketones, which can be sugars too, such as fructose, which are ketoses. So we're going to see that here. And we're going to learn that carbohydrates can be single units. They're just monosaccharides. They're a single molecule of a sugar. They're not connected. They're not polymers that are covalently bonded. But we can also have sugars that are covalently bonded. We can have glucose molecules that are just individual molecules. We can have moles of that, right? But we can also have large sugar molecules that are more than monosaccharides. They're more than just one glucose molecule. They might be two or three glucose molecules, right? So, um, sugars, carbohydrates, we need them. Um, Major source of energy from our diet. They are made from carbon, hydrogen, and oxygen, as we'll see in the structures. Saccharides, that means sugars. It's just another way to say sugars. Remember how I told you chemists love to have four, five, at least three names for everything that you hear of. So there's a lot of synonymous language being used. Saccharides, sugars. Depends on who you're talking to. Chemists might prefer saccharides, but some prefer using the term sugars, right? Nutritionalists always like to say usually carbohydrates. Okay. Uh, Classify, a monosaccharide based on whether it's an aldose or a ketose. And I've already pointed out that aldehydes, sugars that meet the structural qualifications to be a sugar, can be either an aldehyde or a ketone. And when it's a sugar, we don't just call them aldehydes anymore. We call them aldoses and we call them ketoses. And then we indicate how many carbon atoms do we have in that sugar, right? So starting off, we are looking at nature, the environment, how it's all connected. It really is. Plants need CO2. One of the biggest ways China is fighting their pollution problem is more plants, more green plants everywhere, plants on the side of the road. When I was there in China and I got to see all the different plants and trees and stuff that they had everywhere, even on their highways, a very beautiful sight to see. America is also adopting that. We see a lot of that in the United States as well. But what happens here? We have CO2. We can combine it with water and some energy, and then we can do photosynthesis through plants. We can't do photosynthesis, but the plants here on Earth that we nurture, they can do photosynthesis and can make glucose molecules, a carbohydrate, a sugar. And we produce oxygen. So plants are so important to our survival, right? We need oxygen to live. We can't, anybody comes up with the idea of getting rid of plants, they're wrong. I'm joking. But glucose, we can also do this backwards for animals and mammals, and even bacteria and all sorts of organisms, including us. We can somehow break down glucose with oxygen to help, and we can do what they call cellular respiration, and we can extract energy, water, and we end up producing CO2. So this is a cycle, and we see this here how we need energy from the sun to push photosynthesis. Photosynthesis will produce carbohydrates, but to do photosynthesis, we need carbon dioxide and H2O, and of course, the plants. Then we can go ahead and make carbohydrates, and then that energy can be extracted by us mammals or bacteria or who or whatever we might be, and we are able to have energy produced in our body so we can move and exist and perform needed work, like doing your homework. And then we can have respiration, where we give off CO2 and H2O back to the plants, and back to the organisms. Um, of course. So, moving right along, carbohydrates is fascinating. Uh, Guess what? What do you think? You think that sugars and carbohydrates might have some drug properties in how they influence our body? Well, I'm going to tell you that of course they do. We don't like to think of food as a drug, but food is of course a drug. Um, it's not an expensive drug oftentimes. It isn't, depending on what country you live in. Food prices are going up in the United States. But food can be a drug, and sugar, as you can imagine, could be modified to be a drug or a different type of acting drug. So it's not a bad strategy to use sugars to come up with different drug synthetic routes, and chemists do that nowadays. So, but let's look at the very core of what carbohydrates are, and we can look at honey, which contains monosaccharides such as fructose and glucose. So you're talking about the ketose, fructose, and the aldose, glucose. So wow, honey is very lasting, and it's a preservative, a natural preservative. People love it, and they swear by honey as a health remedy. Um, to each their own. Try and things and see what works when there's not too much danger involved in that attempt. Monosaccharides here reacting with enzymes, for example, um, and an acid. And here they talk about how there's no hydrolysis. If we go to molasses, which is another form of sugar, this is maltose, a disaccharide. Okay, so you have 2 units of sugar. Now you have the term disaccharide with water. You have acid and an enzyme. The acid and the enzyme here, unlike on the monosaccharide, can break the disaccharide up into monosaccharides, as we saw from the above, right? So we have all sorts of enzymes. These enzymes are just breaking the sugar bond that's involved. The glycosidic bond, by the way, they call this a glycosidic bond, okay? Um, and if I were to talk more about that, and we will. Um, we'll find out some interesting things. So we look at monosaccharides here. What are they? 3 to 7 carbons long, typically. That's what our bodies might use. We have all sorts of carbon sugars in our body that are 3 to 7 carbons long. Um, we have, remember, the carbonyl group is central to these aldehydes or aldoses, and this carbonyl is central to these ketones or ketoses when it's a sugar. And what distinguishes this as a sugar, as well as having the aldehyde carbonyl group and the ketone carbonyl group, it's all these hydroxyls. Look at these hydroxyls. Everything is hydroxylated. Every carbon is hydroxylated except one carbon. That's the carbonyl carbon. It does not have a OH group on it. So guess what? These are very water-like. Even if they're 6, 7 carbons long, when you add all these hydroxyls to it, it's very soluble. If it was a 6 carbon long chain with no hydroxyls, it would not be soluble. Remember what we learned before, but we put these functional groups of alcohols on here, and ketones, and or carbonyls and ketones and aldehydes. So these ketones are also classified as ketoses, and these aldehyde groups are classified as aldoses when they're sugars and they're highly hydroxylated. There's another trick the body loves to do. The body loves to hydroxylate things itself if they find it toxic. That is just one way the body will try to remove something that is toxic. If possible, it will hydroxylate. What do I mean by hydroxylate? I mean add OH groups. As simple as that. An enzyme that comes on and tries to put OH groups on some type of molecule to get it out of the body. So these structures, if erythrolose, all important, but notice the simple difference. One is an aldose, one is a ketose. They're both have the same chemical formula, I believe, and they both have the same number of carbons and hydrogens. So again, they're structural isomers, but they have different properties. Monosaccharides, let's again just go back to focusing on a single molecule of sugar and what type it might be can be characterized not only by the aldehyde or the ketone group being present, but also how many carbons are involved. 3 carbons here, 4 carbons there, 5 carbons here, 6 carbons here. So an aldopentose, we know from this name, has got to be a 5 carbon. Um, we know it's got to be a type of sugar. It must be an aldehyde group because it says aldo. So we have a 5 carbon sugar that has an aldehyde attached to it. The next one we see is a ketohexose. We know because of hex, we must have a 6 carbon uh, uh, sugar with a ketone group. And of course, it's a monosaccharide here. Okay, we're just dealing with single sugar molecules, not polymer chains of sugars, which are very prevalent in our body, as well as monosaccharides. So we look at some sugar molecules, and 3 of the 4 here happen to be aldehydes, right? 3 of the 4 are aldehydes. Glyceraldehyde, also called aldotriose. Threose, also called aldotetrose. Ribose, also called aldopentose. And fructose, the only ketone shown here, is also called ketohexose, right? So the more common name is the top name, like ribose and threose and glyceraldehyde. All of these are present in the human body, all of them. Ribose, RNA, is everywhere for our ribonucleic acids. It has to have the sugar backbone. Unlike ribose is, of course, the fuel. I shouldn't say fuel. It is a building block as opposed to like glucose, which is more of a fuel to the glycolysis process. But of course, ribose can be pulled off in some kind of what they call metabolic shunt from the process of glycolysis or some other place in the body. But we don't really need to worry about that. We're looking at the big overview. We're seeing glyceraldehyde. You see that in different parts of metabolism in the human body. Enzymes recognize all of these often separately. A different enzyme for each sugar, maybe more than just one enzyme. So the body is quite an exquisite machine that's developed or been developed or however it's happened. It is very sophisticated. Our human body is quite sophisticated. We're learning more and more. Okay, so the question is identify one as aldo or keto, and is it a tetratose, a pentose, or a hexose? Well, we try to answer the first question. We say aldo or keto. That looks like an aldehyde. We don't see a ketone anywhere. Here we see a whole bunch of hydroxyls. We count up the carbons, including the aldehyde carbon, 1, 2, 3, 4, 5, 6. We know that has to be a hexose, right? We look at B, one, two, well, let's try to figure out what it is. Is it an aldo or a keto? Well, we don't really see an aldehyde group with a carbon and a hydrogen and a carbonyl, but we do see the ketone group trapped between or locked in between 2 carbons. 2 covalent bonded carbons are next to the carbonyl group, which means we have a ketone, which makes it a keto. We count up the carbons 1, 2, 3, 4, 5. Guess what? We have that would be a pentose. Okay, so We have an aldehyde and a hexose, and we have a ketone and a pentose. So aldohexose and we have ketopentose. So that is our way to make a very common, if not IUPAC sensitive name as well. And we are done. Thank you. Kim 114, GOBC. We're going to be doing more stuff. I welcome any questions, and you have a great day.